Most of you here today receive our quarterly newsletter, The Beacon. We were so proud in the recent issue to tell the story about Dr. Johnathan Garlick and his collaborative efforts with Dr. Michael Whitfield of the Dartmouth-Hitchcock Medical Center. Johnathan and his team at the Johnathan Garlick Labs at Tufts University work with developing skin samples that can be used for both research and the testing of medications. They have developed 3D-like skin-like tissues that mimic the form and function of the human skin. And we have had Johnathan and his staff into one of our support groups recently to just really give us a hands-on, literal hands-on understanding of how these skin tissue samples work. I'm so pleased to introduce Dr. Johnathan Garlick to speak to you on this topic. Ladies and gentlemen, welcome Johnathan Garlick. Thank you, my friend.
Thank you, Jack.
Hello. Good.
Works. Thank you. Thank you for having me here today. Can you all hear me with my mic on my lapel? You can hear me in the back? Great. Can't hear me in the back. How about now? You can hear me now? Good. I'll talk a little bit louder. So I'm inspired to be here to talk to you about this topic of how we make lab-made skin. And in our research, we are trying to create a skin that has the properties of skin in a scleroderma patient. So I'm going to tell you about that, and I'm going to describe really 4 questions. Actually, it's a good day to do the 4 questions. Today's the last day of Passover, as some of you may know. So these are very different questions. And the questions are going to be, why do we need lab-made skin that has the properties of scleroderma? How can we make it? How can we use it? And how can it help us? But before I start, I just wanted to look at this. As I was sitting there, I was looking up and said, yeah, this tells the story of how my laboratory is supported in its research by the support group at the New England Scleroderma Foundation. When we come to visit in Topsfield, I always bring my lab with me. I bring the people that are actually doing the work in my lab. And I see many friends here in the audience that know them now. And when we come to the lab, when we come to visit you, we get an education. And when we leave after visiting you, we always feel that we've shared some information with you. We tried to update you on our research, but we've drawn a lot of inspiration and education from those interactions. So I come today to give you a research update on what we're doing. And I'll start by sharing with you the skin that we make in our lab. So here it is, lab-made skin. And I have to thank Kathy and Don Legere. Kathy and Don visited me yesterday. Don actually contributed to our research by giving a blood sample, and then I gave them a little tour of the lab, and I said, here, this is how we make artificial skin. And Kathy said to me, that's not really artificial skin, we need a better name than that. So I said, oh, what do you want to call it? And they said, let's call it lab-made skin. So now you're going to see my presentation, thanks to the 2 of you. We are now calling this lab-made skin. So here it is, lab-made skin. We make it from scratch. This is actually a lab-made skin that we made from foreskin cells, which are very easy to access. And I'll be over there on the side, and I'll be able to show you what this lab-made skin looks like. I have another one, skin in a bag over there. But just to demo it for you and to stimulate your curiosity, it's actually made on this little plastic cup that we use to grow the skin in multiple layers, and I'll describe to you how we do that. But just to intrigue you, I can pull off that lab-made skin. Here it is. And I can pull it, stretch it, and most importantly, we can do experiments with it. So come over there later, you'll be able to touch it and feel it. This is completely sterile, so don't worry about holding it before lunch. So imagine if we could make a skin that had the properties of a Scleroderma skin that looked just like this. Why do we need it? How are we going to make it? How can we use it? And how can it help us? So I'll put this here for now and allow you to come over later and actually feel it for yourselves. So the first question is, why do we need lab-made skin? Why do we need an alternative way to do our research? Why do we need a different system to study Scleroderma? Well, what you can see here is that we need more predictive ways to study Scleroderma in the laboratory. And we need to study them in tissues that look like, feel like, and respond like the actual tissues in our body. Now, for the most part, we study Scleroderma in plastic dishes. Here's an example of a plastic dish. Now, plastic dishes are a great way to grow cells. One of the advantages is, as you see here, the cells are easy to use and manipulate and grow. But actually, cells in our body don't grow on plastic. They grow in tissues. And for example, I'm going to do a little experiment for you right now. You hear the sound of the plastic. And this is the sound of our tissues. It's very different. This is plastic. This is flat, hard surface to grow cells. And what we say is that these surfaces do not mimic the complexity of the tissues and organs in our body, so we need to create a system that's going to allow us to closely mimic the tissues and organs in our body. And I'll tell you how we do that. Now, we also use animal models of Scleroderma, and they're great. They're very helpful for certain purposes. Animals have many complex tissue types, so clearly they serve an advantage for studying Scleroderma. But the problem is that a mouse, except in a few cases, a mouse is not a human, and they're not as helpful for studying early stages of disease processes in a mouse as you would study in a human tissue. So I'm going to make the case, as you probably know, that the world is not flat. And biological research should not be flat either. So here we have this plastic dish. We're growing cells on a flat surface. It's very helpful to grow the cells on the surface, but that may not be enough. So we grow cells on the surface. It's a great surface for the cells to grow. We provide them with a broth, a high-octane growth broth that allows these cells to grow. We put in cells that really want to grow, cells like stem cells that we take from patients. But in our bodies, as I've already said, cells do not grow on plastic dishes. They grow in complex tissues. And we need to think about ways that we can make complex tissues if we're going to study disorders that are complex. So let me share with you what our dream would be for building complex human tissues. This is kind of the best case scenario. And then what I'll do is I'll tell you about progress that we're making in building skin using Scleroderma cells. And I'll tell you how far we come and what we're going to be doing with those cells in future research. So you can see here that in order to make a complex human skin, we need multiple components. We need to have a scaffold, which is the internal structure that allows us to have cells that are in relationships with each other. When we put those cells into the system, is there a pointer here? Oh, there it is. Perfect. So when you put these cells into the system, you can see that the cells here are now in that three-dimensional structure. That allows cells to communicate with each other. Cells need to communicate with each other from different cellular compartments. They cannot do that on a plastic dish when they're growing on a flat surface. We can also put in blood flow channels into these tissues that allow cells to communicate as if they were interconnected. We can grow multiple cell types, as I'll tell you for what we're doing with skin. And ultimately, if you want to put in nerve cells, we want to put in immune cells that can actually simulate the way immune cells react in our body. We need to be able to image these tissues to get information from them. And we also need to be able to analyze these tissues. So if we think about how we can, in the laboratory, use tools that allow us to create tissues that have three-dimensional context. The plastic dish only has 2 dimensions. It's growing in this dimension, but we need to think about ways to create tissues that have 3-dimensional structure, so they'll have organization and structure just like they have in our body. I could never lift cells off the bottom of a plastic dish, but here's a real skin that actually has the features and properties of skin in a very similar way to the way it grows in our bodies. So the question is, why do we need to have skin that simulates the way tissues grow in our body? And we plan on using Scleroderma skin so we can develop drugs to treat Scleroderma in a more efficient way. Most drugs that are tested never reach the market. And in fact, basic research starts off with thousands of substances that are then tested in animals, that then go into human clinical trials. And sometimes, if it's found to be effective in human clinical trials, it gets into patients. We make the case that if we can use artificial skin as a way to test and screen drugs, we can move beyond the mouse, not just test in animals, but actually test potential drugs before they go into animals in a tissue that more closely mimics the skin in our body. So the question is, how can we make this skin? Can we make a skin that looks like this with patient cells from Scleroderma patients, and then use that skin to test new drugs in ways that are going to be more informative and more predictive about how they are actually going to work. So first, let me share with you how we actually make this skin. And I showed that to Don and Kathy yesterday when they came to the lab, so I had to bring the lab to you today. The first thing is we want to use as many types of Scleroderma cells as possible. The second is we need to confirm that the skin has features of Scleroderma. The third thing we need to know is, will the skin that we make compare to Scleroderma skin? Is it similar? And the last thing we need is a lot of luck. Tissues are hard to make. The work is very challenging and technical, but we have the expertise to overcome those challenges, and I'll tell you how we do that. So this is the tissue we want to make. This is human skin. If I took a piece of skin from any one of you, put it under the microscope, looked at it from the side, you would see these multi-layer tissues. Here you see the upper layer. Actually, on the surface, you see the protective layer that protects us from the outside world. We have little hairs coming up through it. This is the surface layer, this red material. Then you have a pink material here. That is the deeper layer, known as the dermis, the connective tissue. And that is where we are really interested in understanding the processes like fibrosis that cause the connective tissue to thicken up. Can we make this skin, and how would we do it? Well, here's an example, side by side. If I took this skin that I just showed you, cut it in a section, looked at it under the microscope, I see exactly these layers. I see this surface layer here. This is equivalent to what we're seeing on the very top of the skin. The pink cells, the cells that you see here, these pink-purple cells are what we see here, the epidermis. Underneath it, we have the dermis. Here is the dermis. Here are all the cells that are present in it. So this is a pretty good approximation. This is made with foreskin cells. The question is, can we do the same thing with Scleroderma patient cells? So I'm going to tell you how we do this. Here's the one system I just described with the multi-layer tissue. Here's another system in where we only have the deeper layers of the skin, which we call the dermis, and I'll tell you how we do that and how we apply it. So first of all, what you see here are cells that were actually taken from the skin of scleroderma patients, some of you who are here in this room today. And we can grow them on the plastic dish first. And as you see, if we put down 20,000 cells, within a couple of days we have 100,000 cells, and a few days later we have 1,000,000 cells. Each of these little lines here is another cell type. But I want to say that none of this work happens or would happen without the help of those of you who contributed to this study, and I do mean made a contribution from your skin. So thank you to the Scleroderma Foundation New England for these contributions. We continue to grow these cells. They are banked in our laboratory. We can take them out whenever we need them, grow them, and put them into our skin system. Most recently, we've been able to expand the growth and development of the skin tissues by incorporating new cell types, and this is being done in collaboration with Dr. Michael Whitfield, where we're taking blood cells. Here's a blood sample. Immune cells of interest are called macrophages. They're known to be important in the development of scleroderma. We take these cells, put them on a dish, and this is what they look like. Now, I want to take a moment to thank Jack for seating me at the last patient education seminar with Michael Whitfield. We had lunch together that day, and I can tell you amazing things happened at the seminar because as a result of that lunch, we are now working together. We have grants, work in progress, and a lot of the work you're going to hear about today is as a result of that lunch we had together, and maybe we should have lunch together again today to continue that work. So thank you, Jack. Great things happen at this patient education seminar. So, how do we make this skin? The way I describe this, like making a cake, it's multiple layers. We first take the cells that I showed you from scleroderma patients, put them into this collagen layer. We grow that for a few days, and layer by layer, then we add the surface cells to it. And we grow the cells, as you can see here. They're only moist from underneath, sitting on the feeding gel, and they are dry on top. And when we put these cells in an environment when they're dry on top and fed from underneath, it exactly simulates the way our cells and our skin grow in our body. Dry on top, moist underneath. The cells are very smart. They immediately snap into a program that allows them to make a skin that mimics the form and function of the skin in our body completely in the laboratory incubator. This is what it looks like. Here's our little skins. You can see they're dry on top. And we have skin in a dish that we can use for all of our experiments. The next question is, could we make these tissues even smaller? Because if you're going to screen a lot of drugs, you need to have smaller tissues. So we developed another system, our second system, which has some of the features of skin, but it really only has the deeper layers of skin, the dermis. And the way we do this is we take patient cells, put them into that cup insert, a very small one. We let them make their own connective tissues, their own dermis. This is what it looks like. And what's great about this is we can make them in a smaller format. We can make these into what we call mini tissues. And what we find when we do this, different cells make different thicknesses of tissues. So here you see this is just the deeper layers. This would simulate the part of the skin that becomes thickened in scleroderma. And now we know we have a couple of tools that allow us to actually take cells, put them into these experimental systems, and create tissues that are going to allow us to look specifically at processes that occur during the development of scleroderma in patients. So we asked the next question, now, can we do this with scleroderma patient cells? And that's where our research started, and our collaborations with Michael Whitfield are headed. So here we see some examples of these smaller tissues. We can make these mini tissues, and here's some examples of what that looks like. It's a little experiment we did where we added this compound called TGF-beta, which causes these tissues to become thickened and contracted. And you can see if we have more tissues, we can add more test drugs to allow us to see how these cells and tissues are responding. So in order to make skin that's going to look and feel like this, but actually have scleroderma cells in it, what do we need? We need ways not only to incorporate the cells, but we need to tell that the skin has the features of scleroderma and that it compares to the skin from a scleroderma patient. So I'm going to show you two slides that actually have some data. And what you can see here is when we take the skin cells, grow them in the system, we can see that, remarkably, we were shocked when we saw this result, that cells that are not from a scleroderma patient made a very thin tissue, but cells from a scleroderma patient made a very thick tissue. You can see that this tissue is much thicker than this tissue when we look at it from the side, and we see that represented here. So the first observation was, yes, the thickness of scleroderma tissue is definitely increased. And the second question was, did the stiffness of scleroderma skin actually increase compared to skin that was made from patients that did not have scleroderma? And we use a really interesting tool to test the stiffness. It's like a little probe that we put onto the skin surface. We make the artificial skin, we test it, and what we see is that, yes, indeed, we can see that the skin that was made from scleroderma cells is actually stiffer from the skin that was made from non-scleroderma cells. So now we know we can make the skin. It has the properties of scleroderma. And in addition, when Dr. Michael Whitfield has done some studies looking at which genes are turned on and which genes are turned off in that skin, this is a paraphrase of what you said, Mike, at one of our recent conversations. The way genes are turned on or off in lab-made skin is more similar to patient skin than any other way of growing scleroderma patient cells. Would you say that that's accurate? So the bottom line is, and what I want to share with you, how can we use this skin? Well, I've already mentioned to you that we can accelerate and streamline drug testing. So if a company has a drug that they want to test that they think may potentially improve scleroderma, we can accelerate this drug discovery pipeline. In addition to testing it in animals, we can test it in our skin models. And the way we think about it is that in the discovery process of new drugs, when it goes from early screening into clinical trials, if we can use a scleroderma tissue, we will be able to make better choices about which drugs are going to be effective in humans and which drugs will not. And finally, how can it help? How can it help our community? We feel that using these tissues, we can shift our research paradigm to include research using tissues. And in fact, if we go from basic research to animal models to clinical trials to therapy, we feel we can make the biggest difference towards improving patients' lives by including these tissues in the drug discovery process. So finally, where do we stand now and how are we moving forward? And I probably have a question in your mind as to how long is this going to take? Not just the presentation, but also how long it's going to take until we actually use these. So I can tell you that we can use these tissues even now to study the causes of scleroderma. So if there's anybody in the research community that wants to use these tissues, we can actually make them available and they can be used for understanding scleroderma processes that lead to the formation of scleroderma tissues. We're going to need more time to test and develop ways of using this for drug screening. That's going to take some time. Mike and I are working on a new grant now. We're going to need some more time to figure out how to make these tissues kind of fit that mode. And we're also going to need some time in the future to think about ways of making tissues that are specific for individual patients that could be used to test drugs in individual patients as well. There's a new initiative that we're a part of to take a scleroderma patient tissue and incorporate it into a range of other tissue types. You might have a piece of liver, a piece of scleroderma skin, a piece of nerve tissue, a piece of heart tissue, and you could test the drug by putting that drug in and seeing how it interacts with each of these tissue types and understand whether there might be a toxicity that could be associated with any of the drugs that we're testing. And that's the goal, to say, are these drugs going to work in patients, or are they going to harm patients, but doing it in a way that we can predict what their value is going to be. So what we know is we can make scleroderma cells behave the way they do in scleroderma patients in the skin. We can make these tissues more real by adding more cell types. We can make them with human cells in tissues. We can make these mini tissues. And we know that the lab-made skin is a big improvement to growing cells on a plastic dish. So our next step is this collaboration with the Garlick, Whitfield, Pioli labs, as well as Saldara, and I know that Irena is here from Saldara, to make screening tools for scleroderma, understanding mechanisms of scleroderma, disseminating these tools to treat scleroderma, and helping other research labs understand why scleroderma develops. So a big thank you to my lab, and a big thank you to the Scleroderma Foundation, New England. One of our visits there, we can see we happened to be on Valentine's Day, so we shared some good candy. And thank you, Jack, for the opportunity to share this with you. I'll be over there with skin if you'd like to touch it and feel it.